Hello everyone, welcome back to another session in dentistry and more. Today's topic is oral mucous membrane. It is nothing but mucous membrane which is covering the oral cavity. So we know moist membrane is used to describe the most lining of gastrointestinal tract, nasal passages and other body cavities which communicates with exterior. So likewise, the oral cavity also have this lining which is known as oral mucous membrane. Okay, so let's get into details of oral mucous membrane. So we'll begin with its function. So definition is nothing but mucous membrane which covers oral cavity. Functions, we have various functions: protection, sensation, secretion, thermal regulation, absorption and excretion. So in protection, which separates and protects deeper tissues and organs from mechanical stress, forces and external environment. In sensation, the temperature, touch and pain sensation, and also tongue has taste buds. Reflexes like swallowing, gagging, salivating are also initiated by receptors in oral mucosa. And in secretory function, saliva is secreted by salivary glands and contributes to the maintenance of a moist surface. In thermal regulation, in few animals, considerable body heat is dissipated through oral mucosa by panting. Okay. Which is not very active in human. And absorption function, the certain substances like nitrates are absorbed from sublingual region. We know it used for cardiac diseases. We keep the sublingual nitroglycerin tablets and it will be fastly absorbed to the bloodstream. And excretory function, it excrete certain metabolites. So that's about the functions of oral mucous membrane. This is the important part, which is classification. So it is basically classified as keratinized and non-keratinized, and also we have specialized mucosas. Keratinized means the mucous membrane is keratinized. There is no nucleus, no organelles. It is completely keratinized, which is basically associated with gingiva and hard palate. And keratinized structures, you can see that gingiva and hard palate are involved in the mastication. So it bears the masticatory forces, and it is comparatively hard structures. So gingiva and hard palate, which bears masticatory forces. In non-keratinized tissues, we have two types. Firmly attached mucosa. This is also known as lining mucosa, non-keratinized mucosa. Keratinized is masticatory mucosa. This is known as masticatory mucosa, and non-keratinized is known as lining mucosa. The name itself gives the function. One is to do the function of mastication or involved in mastication. Other one is lining the oral cavity. So lining mucosa, we have two types. That the firmly attached ones are soft palate, lip, cheek and ventral surface of tongue, whereas loosely attached are floor of the mouth, vestibule and alveolar mucosa. And we have specialized mucosa, which is dorsal side of tongue, which has taste buds which involved in taste sensation with the help of various papillae, circumvallate papillae, folate, fungiform and filiform papillae. So that will be dealt in the next session. So that's about classification. It's very important. We have three types. Which are they? One is keratinized and non-keratinized. Keratinized is also known as masticatory mucosa, which is gingiva and hard palate. Non-keratinized is known as lining mucosa, soft palate, lip, cheek, ventral surface of tongue, floor of the vestibule, alveolar mucosa. Everything except gingiva and hard palate and dorsal side of tongue is coming in lining mucosa, and only the dorsal side of tongue is a specialized mucosa because it involves taste sensation as a special function. Okay, so that's about the classification. Now let's see the histology of oral mucosa. There are basically two components. Oral epithelium is made up of stratified squamous epithelium, and this is epithelium, this pink one, and the underlying connective tissue is known as lamina propria. So this is the connective tissue part, this brown color. This pink one is epithelium, which is stratified squamous epithelium. So stratified squamous epithelium. So lots of epithelial types are there. So this is stratified squamous epithelium, and the interface between epithelium and connective tissue is usually irregular. So you can see the interface between connective tissue and epithelium are irregular, and upward projections of connective tissue, which is known as connective tissue papillae. So you can see that this brown color is projected into the epithelium. Okay, projected into the epithelium, it is irregular, which is known as connective tissue papillae. Okay, so this is the connective tissue papillae, the upward projections. Connective tissue papillae. Connective tissue papillae is the upward projections of lamina propria into the epithelium. So these papilla interdigitate with the epithelial ridges. Okay, so this is known as connective tissue papillae, and this part. So this is connective tissue papillae, this projections, that is upward projections, connective tissue papillae. And similarly we have the downward projections of epithelium. So this is epithelium, which is downward. This is just a cross section, that's why I'm saying downward and upward. This is a cross-sectional picture, that is why I'm saying downward and upward. So the top layer will be anyway epithelium, and deeper layer will be connective tissue, submucosa, like that. So the connective tissue papillae, which is this one, and the epithelial ridges. So epithelial ridges and connective tissue papillae is interlocked. Okay, so it is interlocked. So if it is very parallel, if it is like this, there will not be any force between this, and if it is like this, so there will be a mechanical locking between these two. So that is why epithelium and connective tissue are interdigitated between these two using epithelial ridges and connective tissue papilla. Hope you understood what is epithelial ridges and connective tissue papilla. And the structural interface between epithelium and connective tissue called as basement membrane. Okay, so that is basement membrane. So this interface between these two is known as basement membrane. So this is basement membrane, this is connective tissue papilla, and this is epithelial ridges. Okay, so we have epithelium here, pink one, lamina propria here, submucosa, that is a layer which is underlying mucosa. So this is the mucosa, oral mucosa, and underlying mucosa is submucosa. And we have periosteum here, that is outer part of bone, and the alveolar bone. Okay, so that is a oral mucous membrane. So the epithelium, so this epithelium is derived from embryonic ectoderm, and the cells vary from cuboidal to flat squamous cells. This epithelium may be keratinized or non-keratinized depending upon the functional requirement. So we have seen the function mastication, lining, and taste sensation. So depending upon this, sometimes it will be keratinized, sometimes it is not. Keratinized epithelium may be ortho or parakeratinized. Okay, so parakeratinized, which has remnants of nuclei. Keratinization means there is no nucleus, no organelles, completely keratinized. Keratin will be deposited throughout the layer, so there will not be any presence of organelles or nucleus. Whereas in parakeratinization and orthokeratinization, in parakeratinization, pycnotic nuclei will be there, that is, a remnants of nucleus will be present. So the cells in epithelium is known as keratinocytes. So the Keratinocytes which contain a fine filaments within the cells, which is known as tonofilament. Okay, so keratinocytes will always have tonofilaments. This is a tonofilament, this is a keratinocyte. So the epithelial cell which is having a thin fine filament which is known as tonofilament. Okay, so these are fibrous proteins synthesized by ribosomes. So when tonofilaments form bundles which are known as tonofibrils. Okay, the cell of filament which is combined to form bundles which is known as tonofibrils. So tonofilaments are formed in keratinized epithelium only. So only in keratinized epithelium you can find tonofilaments. And bundles of tonofilaments attach to an intercellular thickening with an attachment plaque, which in turn adhere to an oval or circular area of adjacent cell membrane which is known as desmosome. It is very important. Desmosome, it is a commonly asked short note. So you can see desmosome here. Between the epithelial cells there will be desmosome attachment. So it is a attachment apparatus. So the bundles of tonofilaments attach to an intercellular, not intracellular, between the cells, intercellular thickening called attachment plaque. Okay, so the attachment plaque which in turn adhere to an oval or circular area of adjacent membrane known as desmosome. So the attachment of epithelial cells to one another by desmosome, and the attachment of epithelial cell to the connective tissue. So this is the connective tissue, this is epithelium. So this is the epithelium, this is the connective tissue. So between the epithelial cells, so this is what I have drawn here. Okay, so the epithelial layers, we have various epithelial layers, 4 epithelial layers. So between the epithelial layers there will be desmosome, but between epithelium and connective tissue there is hemidesmosome. Okay, so this is hemidesmosome, and we have many layers. I have just drawn 3 layers, we have basically 4 layers. So between epithelium and connective tissue hemidesmosome, but between the epithelial cells, that is desmosome. It is nothing but an oval or round structure between the cell membrane which helps in adhesion. And a basal lamina which separates epithelium and lamina propria. We have already seen what is basal lamina. So the connective tissue, that is the lamina propria. The lamina propria is a connective tissue of oral mucous membrane which has 2 parts, and one is lamina lucida, that is the upper part, and lower part is known as lamina densa. So the lamina lucida is a upper clear part that is in contact with the basal layer of epithelium. So this part is known as lamina lucida, okay, which is basically a clear part and which is close to the epithelium. Okay, that is lamina lucida. Whereas and the lower dense layer which is known as lamina densa, which is in contact with reticular layer of lamina propria. Okay, so the layer which is in contact with epithelium is known as lamina lucida, and the lower layer is lamina densa. So the cells of basal cell layer are connected to lamina lucida by hemidesmosome. So basal cell layer are connected to lamina lucida. So the basal layer is connected to lamina lucida. That's why I am showing here. This is the epithelium and this is a connective tissue. Connective tissue, that is lamina propria. The upper part is lamina lucida, and lamina lucida and epithelium, that is a basal layer connected by hemidesmosome. So while moving on to the epithelial type and its maturation, we have 2 types of maturation pattern. One is keratinization and other one is non-keratinization. Okay, so in keratinized epithelium we have 4 layers, that is stratum basale, stratum spinosum, stratum granulosum and stratum corneum. So I am talking about this pink shaded epithelium. So stratum basale, stratum spinosum, stratum granulosum, and stratum corneum. So this pink is elaborated here. So the stratum basale is attached to the connective tissue by hemidesmosome, and rest of the cells are attached by desmosomes. Okay, so in keratohyalin, granules are present in stratum granulosum. That is why it got that peculiar name, because of this hyalin granules. So let's start with stratum basale. It is a deeper layer which is present above the basement membrane. So this blue line, hope you remember, this blue line is basement membrane, which separates epithelium and connective tissue, or in exact epithelium and lamina lucida. Okay, so it is connected by hemidesmosome. So they are cuboidal or columnar cells, and cells of basal layer has most mitotic activity. So the cell production is happening here, and it grows and it goes through each layer, and it becomes keratinized and will be removed. At the same time, a new cell will be produced at the stratum basale. So this layer is also called as geminative layer because it produces cells. They are attached to the basement membrane by hemidesmosome. Okay, so this is hemidesmosome, this is connective tissue, this is epithelium, this is epithelium, this is epithelium, and this is epithelium. So you need to visualize it in all section. The epithelial attachment is between the epithelium, we have a desmosome. Between epithelium and connective tissue, we have hemidesmosome. Whereas the second layer, that is stratum spinosum, they are arranged in several rows. This is just a picture to differentiate between the cells, not the exact one, which has a large elliptical or spherical shape. Cells are fused together due to the presence of intercellular bridges or desmosome. So desmosome is a very important short note. So these peculiar appearance with the presence of intercellular bridges or the desmosome giving a peculiar appearance which is known as prickly appearance. So prickly appearance is seen in stratum spinosum, and they contain glycolipids and which is originating from Golgi complex. Now we have stratum granulosum. The cells of stratum granulosum are flat and are found in layers of 3 to 5 cells thick. So it has 3 to 5 cell layer. This layer is prominent in keratinized epithelium because it produces keratin fibers, but it is absent in non-keratinized epithelium. So these cells have keratohyalin granules. That is why it got this peculiar name, and it is found in the cytoplasm, and these granules helps to form the matrix of keratin fibers. Okay, so it is involved in production of keratin fibers. And we have the stratum corneum, that is a superficial layer which are flat without any nuclei and full of keratin filament, which is surrounded by matrix. This is completely dehydrated layer. These cells are continuously being removed and are replaced by epithelial cells. So one cell is removed from here, so another cell will be replaced here. So cells will be produced here. So it is a continuum. Okay, one is removed, one is created here, and the total length and total integrity will be maintained. So it is removed from the stratum corneum. So we have two types of keratinization, orthokeratinization and parakeratinization. In orthokeratinization, there is no retention of nuclei, but in parakeratinization, there is retention of remnants of nuclei. Now move on to the non-keratinized epithelium. That is, the keratinized is seen in gingiva and hard palate, whereas the non-keratinized is all the other epithelium without dorsal side of tongue, which is specialized mucosa. So the non-keratinized epithelium, the basic difference is it doesn't have stratum granulosum. So this layer is not there in non-keratinized epithelium. Okay, so it has just 3 layers, that is stratum basale, and the middle layer is known as stratum intermedium. Okay, not spinosum, stratum intermedium. Stratum intermedium, and the superficial layer is known as stratum superficiale. instead of corneum, which is known as superficial. So stratum superficiale, stratum intermedium, and stratum basale. So stratum basale is layer similar to that of keratinized epithelium. Only difference is that this layer is non-keratinized epithelium are slightly larger than that of keratinized epithelium. Okay, so there is no spinosum. This intermedium, we can imagine it as intermedium, basale, and superficiale. And intercellular bridges are less conspicuous. In stratum intermedium, we have glycogen, and keratohyalin granules are very rare because it is not at all present. And stratum superficiale, the cells appear slightly more flattened than other layers, and they contain dispersed tonofilaments and nuclei and are dehydrated cells. And this surface is flexible and tolerant to compression and distension. Okay, so that is the difference between keratinized and non-keratinized. Keratinized has 4 layers, which are the basale, spinosum, granulosum, and conium, whereas the non-keratinized has 3 layers: conium, intermedium, and basal layer. Sorry, not conium, stratum superficiale, stratum intermedium, and stratum basale. So this lamina propria, we have seen the lamina propria. We have learned it has 2 layers, lamina lucida and lamina densa. We can also say that it has papillary and reticular layer. So before we learned lamina lucida and lamina densa. Lamina lucida is towards epithelial ridges, that is a connective tissue papillary layer. Then lamina densa is coming here. Okay, it is a layer which is above or closely associated with reticular layer. So lamina lucida towards the epithelial ridges or connective tissue papillae, and lamina densa is below that. Now again we have this classification, that is papillary layer and reticular layer. So this is a reticular layer. Okay, reticular layer is involved with collagen fibers and blood capillaries. So submucosa, we have the submucosa, which is connective tissue of variable thickness, which serves primarily as attachment of lamina propria to the underlying bone or muscle. So this is the attachment of lamina propria to the underlying bone. So submucosa contain glands, adipose tissues, and vascular structures and nerve components. So that's about epithelium. Now let's learn about epithelial cells. Now let's learn about keratinocytes and non-keratinocytes. As the name suggests, it has keratin, it doesn't have keratin. Keratinocytes, which constitute the major part of epithelial cell. The cells are arranged in different layers, and during maturation they either change to keratin or share in keratin formation. Okay, they will be involved with keratin formation. And these cells are present in sheets and attached to each other by cellular junctions, and cytoplasm contains tonofilaments. So I told you tonofilaments are always associated with keratinocytes. Whereas non-keratinocytes, this thing we already learned in our gingiva, and I'll just recap the non-keratinocytes. They are Langerhans cells, Mucous cells, melanocytes, and lymphocytes. They present in both keratinized and non-keratinized epithelium and appear as clear cells in H&E staining, and they are present as scattered and not in sheets. Okay, so there will be a clear halo around the nuclei and there is no cellular junctions. They do not play any role in keratohyalin granules or keratin formation. The first one is Langerhans cells, which is found in stratum spinosum and occasionally in stratum basale. These cells have long, thin extension of cytoplasmic membrane, which is known as dendrites. They can be distinguished from keratinocytes by the absence of desmosome and tonofilaments. So desmosome and tonofilaments are only for keratinocytes. Langerhans cells are seen in stratum spinosum and basale. And they can be distinguished from melanocytes by the absence of premelanosomes. They contain the rod-shaped or racket-shaped Birbeck granules. Okay, so that is very important. Birbeck granules, which is seen in Langerhans cells. Birbeck granules. We already finished this in gingiva. Birbeck granules. They are antigen-presenting cells, which allows positive identification at the ultrastructural level. They engulf antigens from the external environment, and intracellular lysosomes split the antigens into peptide components. These fragments are then transferred to T lymphocytes. So that is about Birbeck granules, which is important short note. Now let's move on to the Merkel cell. Merkel cell are situated in basal layer of the gingival epithelium, basal layer, which possess desmosome and tonofilaments. They are usually associated with axon terminal, that is nerve ending. So it is associated with touch receptor. They are usually found in groups. Melanocytes are melanin producing cells. They are located in basal layer of epithelium. These cells arise from neural crest cell, lack tonofilaments, desmosome and hemidesmosome, are highly dendritic in nature, and most characteristic feature of melanocyte is the melanosome granule found within the cytoplasm. A more heavily pigmented gingiva is due to the production of melanin and its subsequent uptake by the epithelial cells. Lymphocytes are inflammatory cells. They are with larger nucleus, no desmosome or tonofilaments. They are involved with inflammatory responses. Okay, so in next session I'll be explaining about the keratinized mucosa, that is gingiva and hard palate. Gingiva we already finished in a long session it was. So gingiva and hard palate are keratinized mucosa, and after that we have specialized mucosa about tongue and its papillae. Okay, so that's all about the oral mucous membrane part 1. So in part 1 we have learned more about its epithelium, its functions, its layers, lamina propria. So there are lots of questions will be asked: lamina propria, lamina lucida, lamina densa, papillary layer, reticular layer, desmosome, hemidesmosome, keratinization, non-keratinization layers, stratum basale, stratum spinosum, stratum granulosum, stratum corneum. In non-keratinized, stratum basale, stratum intermedium, stratum superficiale, and keratohalin granules, Birbeck granules, Merkel cell, melanocytes, Langerhans cells, and epithelial ridges, connective tissue papillae. So the lots of questions will be asked from this session. Okay, so next thing I'll be explaining about the masticatory mucosa and lining mucosa. Thank you.